So now we are going to shift to the proof of concept demonstrator. As I told you briefly this morning, we are going to open a call dedicated, and we are going to offer access, free access to integrators. So we partners, we are very pleased to partners with IVEth, with ATGT and MEARY Center. So we are going to offer this free access to expertise and equipment. So now it's time to discover, to understand more what they can propose to you. So this way you can also take the time to discuss during the launch time. They are going to be present in the village. So it's time to know more about IVEth first, please.
Hello everyone, my name is Galy Aubertin. I'm the Chief Scientific Officer of the IVEth integrator, which was the first one that was fully dedicated to extracellular vesicles and also called EVs. So we are particularly focused on the production, engineering and characterization of extracellular vesicles for therapeutic, of course, purposes and also diagnostic. And so thanks to 15 years of experiences working on EVs and an interdisciplinary team of about 10 persons dedicated to the integrator, we build a park of equipment of about 15 equipment dedicated to extracellular vesicles and to the design of this workflow from bioproduction to machine learning multimodal statistical analysis dedicated to quality control. And so we have systems to produce extracellular vesicles, a major one being induced by turbulence flow that is generated a lot of extracellular vesicles. This technique was designed and patented by the lab and licensed to Eversome startup. We have one system that is that was founded by the team BioConvS for la santé. We have strategies to bioengineer either the cells prior to bioproduction or the EVs during or after the production. We have systems to follow the cells, their viability, their number, or the EVs, the concentration and the size during the bioproduction process. We have systems to isolate extracellular vesicles, one preferential one being the tangential flow filtration. That is scalable and quite efficient. And then we have a big park of equipment dedicated to multimodal characterization of extracellular vesicles. So we have systems to follow, to characterize the physical features of EVs, the size and concentration. We have systems to get the phenotypic signature of EVs. We have a system to do a super resolution microscopy to get the morphology, the size, and also phenotypic information at the single EV level. We have systems to get the global biomolecular signature of EVs by Raman spectroscopy. We have systems to separate and analyze the different fractions of the secretome using multi-detector based on optical parameters. And we have systems to evaluate the biological and therapeutical potential of EVs. And all the data that are collected during this, with this multimodal toolbox and also during the bioproduction are going to be put together and to be analyzed using machine learning tools in order to design new quality control testing method in order to say that a batch is compliant and can go to the clinics or not. So thanks for your attention. If you want to know more about IVEth or extracellular vesicles, please come and see us. And by us, I mean Faima De Federico, who is in the room here, and also Sarah, who is in the back here, and me, of course. Thank you.
Thank you a lot, Kelly, for this overview. Now, I invite you, Anne, to present ATTG integrator. Thank you.
Yes, I would like to present the Accelerator of Technological Research in Genomic Therapy to accelerate the translation of cell and gene therapies towards the clinic. So our field is cell and gene therapy, and I just would like to remind you very quickly that these types of advanced therapies constitute approximately already 1/3 of the approved drugs on the market in the form of stem cell-based therapy or CAR T cells. But there's many new products that are coming up. You have CAR T cells in sectors other than oncology, or such as in autoimmune diseases, or in fibrosis. You have other immune cells which are being engineered, such as CAR macrophages, B cells, etc. And you have also stem cells-based therapies to treat diseases other than blood disorders. The current technologies are usually lentiviral vectors or CRISPR to genetically engineer these cells, but there are additional technologies coming up very, very rapidly to complement and provide additional mode of action, such as, for instance, prime editing or derivatives or base editing. And also the vectors are changing with targeted envelopes for targeted delivery or also for in vivo delivery. So there's really a lot of things to do in this field. But the challenges are still to try to master the efficacy and the safety of these products, which are usually integrative vectors. So they go into the genome, and so they have a risk of insertional mutagenesis. There's also a very high complexity of the bioproduction process, which also increases the cost of this field. And there's a huge translation gap between researchers and the application in clinic or in industry. And now we're also facing a question of access, where there is a part of these drugs which have no market access. So in that context, INSOM has created the Accelerator of Technological Research in this field, and our mission is to promote the excellence of research in this field by providing, in fact, and ensuring the continuity of the flow of research from R&D all the way to pushing this new concept to clinical trials. And we have this label, which is the integrator label that was provided to us since 2020. To really provide a quality environment and also with the quality assurance that allows us to work with private partners or startups and to help move to the clinic. So we have research projects, and in particular we work on novel immunotherapies, and we are particularly interested in immunotherapies that can be used in the context of frequent disease such as infectious diseases. So I won't go into that for the sake of time. And we also support preclinical activities, and in particular IND-enabling studies that will allow you to evaluate either the safety of your product or to improve the process of production of the product or to validate certain concepts. And one of our key attributes is to be able to, in fact, produce lentiviral vectors to industrial scale and develop processes for the production of the vectors that you want to use, in particular associating high level of purification and concentration by chromatography, and also all the characterization that goes with that in physical particle, infectious particle, composition, measure of impurities as well. So, to have a process that could be transferred to industry. So we provide some service activity in the high value activities that we have, either in bioproduction development, some of the analytical tests, titration, vector copies, impurities, whatsoever. But also we really help people think about how they develop their product and provide some IND-enabling advice and custom development as well. And so right now our service activity is really centered around all of the lentiviral vector, in terms of production, QC, in particular also the genomic analysis of insertion sites. But we are moving into genomics and genome editing safety evaluation through the biocluster Genoterre in collaboration with our colleagues at Imagine and Geneton. And so with that, I just want to finish by saying that we are located in Corbeil-Essonnes, in fact, just above ISTEM in this building. We're a small group, relatively small group, but happy campers. And so you can see two of our posters, I think 19 and 42, I think, here, as well as the stand upstairs. And please contact us. You can check our website as well. And also, I thank all the people who have funded our projects and you for your attention.
Thank you very much, Anna. MEARY Center, we are pleased to welcome you.
Hello everyone, my name is Hélène Boucher Pilet. I'm a pharmacist and I'm a part of Cell and Gene Therapy MEARY Center team. MEARY Center is located in Paris in Saint-Louis Hospital, and MEARY Center is led by Dr. Fabregat and is a GMP facility authorized for experimental ATMP manufacturing. Our 800 square meter premises include a Class A and B area production and a quality control laboratory. Your activity is focused on the production of experimental ATMP batches, particularly in early stages clinical trials. We are currently running 2 HPAP sponsored clinical trials with MSC and 2 industry-sponsored clinical trials with Start Immune startup. In the near future, we are 6 clinical trials will be set up with CAR T-cell, mesenchymal stem cells, and regulatory T-cells. And MEARY Center has integrator industrial labels also. Our collaboration with team BioConvS has enabled us to acquire a clinical grade cell sorter, the Taito from Miltenyi. And this equipment will be used in 3 phase 1 clinical trials. And the aim is to evaluate the safety of the autologous human regulatory T cells in the prevention of the kidney, liver, and lung transplant resection. And this project is led by Professor Miara in Hôpital Pitié-Salpêtrière in HPHP. My colleague Caroline de Oliveira and I will be happy to discuss it further with you.
Thank you a lot. And to finish, we have the pleasure to welcome you, Stéphane, to tell us more about the Paris Biofoundry.
I'm going to rapidly present you what is Paris Biofoundry. You already, for those of you that were there in the morning, some words about it. Paris Biofoundry is focusing on engineering biology, meaning its platform dedicated to biological engineering. This audience is focused on health, but we are going into all different aspects. In the future, the next industrial revolution, powered by the convergence between biology and other sciences and computer sciences, will allow to transition our production and enable to make most of our goods with biology in all sectors, from materials, energy, health, foods, digital industry. It has a major predicted economic impact in the future. And to enable this revolution, we need dedicated infrastructures that have started to emerge in different countries in the world, mainly in the US, in the UK, and Asia. Europe is lagging a bit behind, so we decided to create the first Biofoundry in France. It's a dedicated infrastructure that is fully automatized, so we remove the human from the experimental aspects, and we use machines to make all the experiments. Our aim is to give access to these high throughput engineering to train a new generation of synthetic biologists, to facilitate both fundamental, translational, and applied research, and to be a driver of innovation and a base for this bioeconomy through industrial partnerships, innovation programs, and creation of startups. So this Biofoundry is unique in the fact that it's a distributed foundry. It's located on 4 different sites. The central foundry, located at Sorbonne University, is making DNA, very high throughput DNA, fully automated, and engineering microbes, focusing on bacteria, yeast, and microalgae. The mammalian Biofoundry, developed in Institut Curie, focuses on engineering and testing of mammalian cells. We have a cell-free Biofoundry focused on cell-free processes that are emerging a lot in Institut Michaelis. And the 4th site is the Genopole, who is focused on scale-up and process development, which is very important for transition to the industry. So I will not detail all this. I invite you to come. We have posters and the people working at the Biofoundry could explain you all the details. But basically, we have 3 major platforms. One is a DNA foundry that is fully automated, a microbe foundry that is also automated, and facilities for proteomics, metabolomics, high quality mass spectrometry platforms, and also DNA sequencing. And this is coupled to bioinformatics and AI. So this DNA foundry takes your designs, what you want to build, and automates the whole process, and outputs hundreds of plasmids per day that are fully sequenced, and there's no human intervention at all from the design to the output. Everything is automatized. The microbe foundry is also fully automatized. It takes the plasmids coming out of the DNA foundry, put it in your favorite organism, and then screen this and automates the phenotyping. We have complementary phenotyping with cell sorting, proteomics, metabolomics, and a number of things we can do. So you input plasmids and you output engineered strain and a huge phenotype dataset. The mammalian Biofoundry is built on the Biophoenix platform, which is a high throughput phenotyping platform in Curie Institute, and will be expanded with engineering capabilities by 2026. The project will start a bit later than the central foundry. The cell-free foundry at Institut Michaelis is using artificial intelligence to improve biosynthetic pathways and improve enzymes. This is powered by the team of Jolou Follon, and has demonstrated that they can improve by several folds different processes like protein production or CO2 fixation in an artificial pathway, for example. And the Genopole foundry is focused on downstream processes and scale up with a huge facility that allows to test all the process from microliters up to liters, and in the future to hundreds of liters, and partnerships with industry to go beyond the cubic meters. All this is not possible if you've got a very strong digital infrastructure. So we have developed a tool which is called Galaxy SynBioCAD. So it's the AI-powered system that enables all we want to do in synthetic biology, pathway design, retrosynthesis, metabolic modeling for the design phase. It gives instruction to the robots to make the engineering, and then analyzes the output of the engineering to improve our prediction model, which is one goal of a Biofoundry. So this AI-powered system will be deployed on all the different sites. And finally, beyond having a large set of robotic instruments and nice software, what we will offer to the community, this will be open in 2025, is high throughput design build test learn cycle, very high throughput cloning and DNA assembly, but also design and testing of genetic circuit, enzyme, metabolic engineering, synthetic pathways, and coupled to a scale-up and process optimization facility. Our mission is also to develop new synthetic biology tools, new molecular tools, but also new software and AI-driven engineering methods. And so this infrastructure is open to academic labs, industrial partners, and startups through either collaboration and services. And we are ramping up now, installing all the instruments, and should be operational during 2025. And I missed it. And we are creating a number of companies. You heard about Generale this morning, which is from the teams that are include that are associated to the Biofoundry. I'll give you two other examples of spin-off of the Biofoundry. BioMeMORY is focused on storing that digital data on DNA, so it's a completely different field. And BACTA is working in the field of materials, of biomaterials, making sustainable raw materials. So to give you a large overview of the activities of the foundry. And we are, of course, not of course, but we are supported by a number of national and regional institutions, and a very strong support from the DIM BioConvS and the Région Île-de-France as a whole, together with Sorbonne University.